Okay, so it's great to be here. As usual, this is one of those very special institutions that's been built up over the years where we try and get people from many disciplines to be interested in various aspects of quantitative biology. And one of the difficulties in this is, of course, we have mixed audiences. And since I've just walked in, I'd like to get a brief background of where people are coming from. So how many of you are trained in physics or mathematics or related areas? Okay. How many of you have primary training in biology? Okay, so about 1/3. Okay, great. So what I'm going to talk to you about, I'm going to lecture mainly on the board. Maybe in the second session, I'll show you a few things on the slides. So one of the most important sort of tools, frameworks, techniques that has become central to modeling in biology is this idea of stochastic process. And I know that you've already had an introduction to this. Vijay has spoken about this before, but I think it's always good to see the same thing many, many times from many, many speakers so that it sort of percolates and internalizes. So I'm going to do this my own way. So a very complicated word. Actually, I don't like it very much, stochastic processes. It's not a common word you use in English. So let's start with things that people probably know and understand, maybe even learned in school. We all know about random numbers. Very easy. And if you've done even a little bit of programming, you know that your computers come equipped with random number generators. These are little black boxes. You say, give me a random number according to some distribution, and it'll spit out a number. So let's first try and understand how that's done. Once you unpack that, you'll start to see what the connection is to stochastic processes. So how many of you have used a random number generator on a computer? Probably all of you. What kinds of random number generators do you typically get built into many languages? Uniform, yeah, that's a good one. Often you have Gaussian or a normal distribution. Sometimes you have more sophisticated ones, but let's just start with those two. Okay. Now, what I'm going to do is tell you that a computer doesn't come built in with these random number generators, but the black box contains something on the inside, right? So suppose I only tell you that the computer is allowed to do a coin flip, right? A heads or tails. And that's the primitive random number generator it comes equipped with. How would you use that, for example, to make a uniform random number generator? Any ideas? How would you use a coin flip? So this is a head or a tail decision to make, for example, a uniform or, for example, a Gaussian. It's easy. This is the easy part. So, yeah. Yeah? Like that. So the first coin flip gets you into this, and the second one sort of splits you up here. And then the third one flips you up some more. Very good. So that's a great way to think about it. This is exactly the same process, by the way, as generating, let's say heads is 1 and tails is 0. Okay? And you pick a certain number of bits of precision you're interested in, say 16 bits or something like that, right? So what you'd like to do is generate 16 random numbers, each of which is just 0 or 1, because that's what the computer can do. And when you interpret this as a binary number, and say divide it by 2 to the 16, that exactly achieves this procedure. Okay, it drops you into a single bin. You can think of it as a comb. And every time you run this algorithm, you generate a 16-bit string. You interpret that as a binary number. You drop it in here. And in fact, that's how you get a uniformly distributed random number. Fairly straightforward. So this is done. How would you get a Gaussian? Also pretty straightforward. Okay, so you're going all out. Give me something, that's true actually. Okay, so if you have this one, you can do many other things. Okay. In fact, if you have uniform, you can simulate a coin flip. And so many of these things move back into each other. But it is a correct answer, but I'm looking for a direct way to go from coin flips to Gaussians. Sum them up, yeah. Sorry, you were saying. You do, yes, you have to do it a large number of times. Yeah. So now we make use of the central limit theorem. And let's say every time you have a heads, you do a +1, and every time you have a tail, you do a -1. And instead of interpreting this whole thing as a binary number, you simply add up the number of ones in now a much larger string, okay, a very, very large string. I'm not saying this is how it's done in the computer, but I'm saying it could be done. And when you do that, because of the central limit theorem, eventually you're going to end up with a number with mean 0 because you're adding pluses and minuses with equal probability. And the variance is going to be n, and the standard deviation will be somehow proportional to root n. And then you can appropriately rescale it the same way we rescale this by some denominator. You can rescale this by the expected variance and bring it back down, and then you get a Gaussian distributed random number with mean 0 and variance 1. So this is all correct. Now what I want to explain is something that's happening here that's slightly more interesting than what we just said. It is the following thing. I can actually imagine, so this is the number of flips, the number of times you keep using this coin flip and adding up the number of ones. Now instead of just being interested in what happens after a large number of flips, so this vertical axis is, let's say, this variable, let's call it x, plus and minus. So what we've said is after a large number of flips, the sum of the number of ones minus the number of zeros is a Gaussian, and that's fine. So this is basic school level probability distribution. But it's actually interesting to ask, if you start off at 0, how do you get there? So you keep a cumulative total of the flips. And every time you do a heads, you go up, and every time you do a tails, you go down. And typically, that's not going to be a smooth curve. It's going to be some jagged thing like that. Okay? This is also something you're familiar with. So this is called a random walk. The process of generating a Gaussian using flips of heads and tails, if you look at it over time, it generates a random walk. And if you do it you start doing it again, you don't get the same curve. Typically, you get all kinds of curves every time you run this procedure. And this is really the beginning of going away from thinking about random numbers, which are just probability distributions over 1, 2, 3, or some smallish number of variables, to stochastic processes. So for me, a stochastic process is a black box that doesn't generate a probability distribution or doesn't generate one number out of a probability distribution. It generates a whole curve. Okay? You can think of it as a random generator, but in the space of curves, which, as you can imagine, is a much bigger and more complicated space than, let's say, a one-dimensional space of real numbers or a two-dimensional space of real numbers. Okay? Are there any questions at this point? So this is the starting point for today's lecture. We're going to think about how, in general, you can have a black box that generates an entire curve according to some underlying law. It could be modeling a physical process like diffusion. It could be modeling a biological process like genetic drift, the number of alleles. It could be modeling a biophysical process like the number of molecules in a cell. So there's many, 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 many cases where this type of idea is very important, that I want, instead of number of flips on the x-axis, I have time. On the y-axis, I have some state variable, and I want to generate these curves. Now, some of this I think you saw yesterday, and Vijay has already started to describe this. So I'm not going to go into too much detail about how to solve systems like this, but I want to explain some of the philosophy over here. How do you generate a curve? In this particular case, it was actually straightforward because time was discrete. It's very, very straightforward, right? So at any point in time, the value of x was literally the number of heads minus the number of tails up to that point in time. And let's say I had a certain number. Of course, I've drawn this curve in a wiggly way, but it would be a step-like curve, right? In this particular case. Okay. So if I know where I am at a certain point in time, then I use this procedure to get to where I want to be at the next point in time. And in this particular case, time is discrete, and the size of the step has to be either +1 or -1. So it's sort of reasonably straightforward. Now let's start to do something more interesting. Okay, I'm going to leave this alone. Instead of doing coin flips and this sort of random walk in discrete time and discrete space, I'm going to do another process which you are all familiar with, hopefully, which is called diffusion. When people say diffusion is a process, they mean it's a black box, which, once you give the initial condition and say go, it will generate a squiggly curve. And every time you run the process, you get a different type of squiggly curve. So just like the heads and tails random walk is a stochastic process, diffusion is a stochastic process. It's stochastic because you don't get the same curve twice. So now, how many of you are familiar with diffusion? Good. So one of the most important things about diffusion is something strange where the total distance or expected distance you travel from where you start doesn't scale with time, like a car or somebody moving at a constant velocity. No, it's scaled with the square root of time. That's one of the things you remember about diffusion. It's sort of a slow thing, right? And this is the extent to which I was taught about diffusion, even as an undergraduate. Okay, and you've all seen that experiment with, say, potassium permanganate, where you put a drop of some dye in water, and then you see the spread of that dye. And if you actually plot the profile of the dye in X, Y, or Z coordinates, or as a function of radius, right, you will actually get a Gaussian like this, except that this becomes the square root of time. So you've all seen this? Is this fine? Okay. Now I want to ask you, so this is baby stuff. How would you generate a curve corresponding to the position of an actual particle, one particle in that diffusing cloud of potassium permanganate? Not the average over all particles, but a single one. That's the question I'm asking you. Any ideas? How would you generate a particular curve? I'm not interested in this Gaussian. That's fine. I mean, there is a Gaussian and it is, but that has something to do with the sum total of what all the particles are doing, or in some sense, an average, right? But how would you generate one curve? Any ideas? If you've seen it before, I'd still be interested in hearing how you've been taught how to do this. Yes?
With computer methods, you cannot generate something continuous. So there is an approximation.
Very good.
And there is a linear approximation between your steps, and what you do randomly is the time that at which you are jumping. So you start and randomly generate your time.
Yes.
And randomly say, I'm going up or I go down.
Okay. And so I'm going to make you pause over there and just latch on to a couple of important things. So unlike the heads and tails random walk, two difficult things have happened. Okay. One is the time has become continuous. So there's literally infinitely many points over here. The other is that space has become continuous, which is less problematic, but it's also, it's a little more complicated. So when I say generate a curve, you can come back and tell me it's impossible for any computer or any finite object to actually generate the curve. It's not a fair question. Okay, so obviously when I'm saying generating a curve, I say, let's pick some sort of discretization of time and some sort of discretization of space. This is what you also do when you solve ODEs. When a computer spits out the solution to an ODE, it's not an analytic solution. It's on some sort of discrete comb of time. So what I'm going to do is to say, well, let's say this is t equals t0 right here. Let's call this t1. Let's call this t2. Let's call this t3. Or whatever. So you tell me a certain number of time points at which you would like to sample the curve. Okay? In fact, you give that to me. as a specification ahead of time. You say, I'm looking at a diffusing particle, and I'm not going to make you work infinitely hard. I'm just interested in where a particular particle in an experiment would be at T0, T1, T2, and T3. And of course, every time you run this, the particle is going to be at different locations. So the variable, this is a sort of compression of notation. X0 means where the particle is at T0, X2 is where the particle is at T2, and so on and so forth. What I'm actually asking you to generate are these three variables given this one. Okay? I'm asking you for the probability distribution jointly of x3, x2, x1 given x0. So what's a curve? A curve is actually a large number of points. Infinitely large, but you need to pick some finite number. Here I've picked a very small number, 3. In practice, you'd pick thousands, but I don't have space and time to write that on the board. So in fact, we come back to the idea of random number generators. So it's a way of thinking about stochastic processes which are just joint distributions of a large number of variables, but each of these variables corresponds to the height of the curve at different sample points. It's another way to think about stochastic process. It's an operational way to think about it because in practice you cannot do infinite and infinitely weakly curves. Okay, so that's excellent. Now, I give you this specification. I want to know where a particle would be if I know it starts somewhere at t equals 0, which I call x0, and I fix it. Where is it going to be at x1, x2, and x3? Okay? And I give you its diffusion coefficient, d. And I want you to generate this. Now, how would you do it? Now it's a bit more straightforward. So I start off over here. Where is the particle going to be at x1? You need to tell me a single number. I don't want a distribution. The given particle, where is it going to be at time t1? So what would you do? So I made you pause in the middle, so maybe you can restart.
So I guess I'm going to say a strange word. The process is memoryless. So at each point, you repeat the process, right? With less of a time. So you can work it up from the distribution at that point.
Right. So let's not, okay, this is a very complicated curve, but I'm going to leave this picture, but now I'm going to start with a clean slate. I know I'm here. This is T0. I want where I'm going to be at T1. And where I'm going to be at T1 is a single number because it's a single realization. It's a single curve whose value I'm sampling at T1. So the way this is done is fairly intuitive. What you do is you say, suppose I knew this probability distribution, x1, which is at T1, given x0, which is at T0. Suppose I knew from some underlying physics, as you do for diffusion, that if a particle starts off somewhere at T0, after millions of iterations, I get a probability distribution of where it's going to be at T1. So now it's easy. All you do is you take this distribution, which in fact happens to be a Gaussian, centered at x0 with variance or standard deviation somehow square root of time. There is a sort of expected distribution here, and you just sample the number from that. And how do you sample the number from a Gaussian? Well, like this. Right? So that's it. So now we have this and this. We don't know where the particle has been in the middle. It's been doing something. We don't care what it's been doing because you didn't ask me for that information. Now what do you do at T2? Well, at T2 it's also straightforward. What you do is you say, now that I've sampled at T1, I do the same thing again. I know that the conditional distribution at T2, given that you are at x1 at T1, is another Gaussian, except now it's been shifted up a little bit because already you're a little bit biased from the previous part of the curve. And then you pick up another point from that distribution. Not the peak, just some other point. Right? And in this way you keep on going every time. And these time points do not have to be uniform in time. It can be anything. Right? You pick another point, and so on and so forth. So now we have a recipe. we have a recipe to generate a curve at any desired sample point. Right? And eventually, you know that the actual particle, we don't know what it's been doing in the middle, but it would have gone through all these points. Right? And so these are the samples that you return to the user and say, that's what you asked me for. And theoretically, you could make the time points finer and finer and finer and finer. And you will converge onto some sort of theoretical, highly squiggly curve, which is called diffusion. Now, here's an interesting thing, which I think Vijay covered as well. Suppose I was not interested in time point t1. Suppose I was just interested in time point t2. Right? So, one way to do that is, of course, I could generate time point t1, and then use that to generate time point t2, and then just not return the value of x1 to the user. You didn't ask for it. But it turns out that I can just give you the direct value of x2 by knowing x0. Okay, this is actually highly counterintuitive. Okay, this actually is a bit strange. When you solve an ODE, for example, the way you find the value of the variable at some large point in time is you have to find all the first few points. Right? There's no way to sort of jump to where, unless you have some sort of analytic expression, there's no way to sort of jump numerically. But stochastic processes have a sort of magic trick where you can sample more and more finely here, if that's what you want, or you could just jump over. And the fact that these two things have to give you the same answer is expressed in one of the sort of founding equations of stochastic processes, which is this Chapman-Kolmogorov equation, which I think was also mentioned. All that, it's a very complicated equation. I don't want you to get worried about it. All that means is the user can tell me what time points to sample, and I can give her exactly those points without having to sample even more finely. Any questions about this? And the way you do that is by generating things like this. Okay, for a Markov process, by the way. Of course, it has to be memoryless, or else you have to store many things. So I'm going to be speaking today about Markov processes. So let me summarize what I've just said. Stochastic, yes. Yeah, so it's a bit of a magic thing, right? So her question was, for stochastic processes, for ODEs, people are always worried about making these infinitely small. You have to go down to a certain step size before your numerical accuracy reaches the level you need to make predictions, say of a plane flying or something like that, right? But somehow, for stochastic processes, you have this little bit of magic where you can leap to a certain time point without worrying about all the things in the middle. Now, it doesn't come for free. That power doesn't come for free. It comes from the fact that you can derive these sorts of objects. These are formulas, typically analytic ones, otherwise can be given numerically, but these are objects which say, I know the distribution at some later time point given where I'm going to be at an earlier time point. And for a Markov process, these objects have to satisfy very strong constraints, which is the Chapman-Kolmogorov. But roughly it means that I can fine-grain the time, or I can coarsen it, and I still get a consistent form for these things. Okay? In other words, by the way, so if going from here to here, the standard deviation would be square root of t1 minus t0. Going from here to here, the standard deviation would be square root of t2 minus t1. Right? It actually takes you about 2 pages of scribbling to work out that therefore going from here to here, you get a standard deviation of square root of t2 minus t0. It's actually a worthwhile exercise to see how complicated it is to work all that out. Anyway, the point I'm trying to make here is, even if one is interested in curves, and even if one is interested in programming a computer to spit out a curve, you first need to be able to calculate these quantities, which are good old-fashioned probability distributions, which basically say, conditioned on where you start, where are you going to end up if you repeat the experiment millions of times. Okay, so although stochastic processes focuses on curves, the little devices it uses to make those curves are all good old-fashioned probability distributions. Okay? This is something that I was never taught while learning stochastic processes. I somehow got, people keep talking about the curves, but then whenever we did the analysis, we kept calculating these things. Right? So the full picture for stochastic processes only makes sense when you think about having a computer and pencil and paper. You use pencil and paper and a lot of cleverness to calculate this, and then, therefore, you use a computer to make these curves. Okay? Any questions about this? So a lot of the calculations you would have done in Vijay's tutorial are devices, tools that help you generate these kinds of objects. Any questions? Okay, good. So now let me move on. This is all very general. So now I want to focus down on a particular stochastic process, just by way of example. Please.
Central theme, and if we do not have the means to generate a PDF, then
you don't have to be, okay, fine. So for example, there are Monte Carlo methods that numerically allow you to make curves. Okay? But all of them involve repeated use of random number generators. Okay? So although it may not be so direct as to have an analytic PDF from which you sample, any method to generate a random curve must involve iterated use of some primitive random number generator. Okay? Good. For diffusion, actually what's happening in some theoretical sense is this underlying yellow curve. Okay? That's the thing we're interested in. These are all tricks to sample that curve. Okay? In the span of our limited minds, and the way we do that is we pick a finite number of sample points and we calculate these conditional probability distributions. Okay? But really what's going on is there's some yellow thing underneath, which is truly what one is interested in. It's a physical process. Something is really going on. Okay. So now I'm going to focus on a particular stochastic process. Put a piece of chalk here. And maybe spend, I don't know, about 15-20 minutes on this before I get to the meat of my talk. So chemistry actually falls into this realm of stochastic processes, which is again something you saw yesterday. So let's zoom in on one particular fairly important process in living cells, just to bring some biology into it. So this is the process of making mRNA. So there's a gene. How many of you know what a gene is? I hope that level of stuff. Yeah. How many don't know? So here when I say gene, I mean just a bit of DNA that encodes a protein, not something more complicated. So somewhere in the cell, you have a gene, and typically the biologist will draw a diagram like this where there's something called a promoter upstream, and the gene will make this messenger RNA. So this is DNA. This thing is called the promoter. So this is the process by which the cell decides to express, it's called expressing, some subset of the proteins that it can actually encode. And it's a huge field of biology to figure out, under what circumstances some particular gene is expressed. This is the kind of thing which John is going to talk about later, right? And everything that happens in your body ultimately comes down to the idea that different cells express different subsets of genes. That's why your eyes are different from your fingers and your heart. But it's not that simple. In fact, the entire process unfolds when you have a single cell that becomes two cells and becomes an embryo and so on and so forth. So the process of gene expression is dynamic. It's not just there are different cell types, but each cell type in its history went through all kinds of transitions. Now, I'm not getting into that level of complexity, but just to say that it's important and interesting to track the number of mRNA molecules that you have as a function of time in a given cell. Okay? So let's again make one of these plots. So let's see what this is going to look like. This is time.
So, Professor, just
Yes.
in that diagram, there is an invisible actor which goes from gene to mRNA.
I'm going to unpack what cartoons like this mean. Okay, so so you see something like this in a textbook, and in principle, it's a recipe. It should be as informative to a biologist as saying diffusion to a physicist, because saying diffusion doesn't just mean diffusion, but it means square root of time, it means the stochastic process, it means I'll be able to generate curves, and so on and so forth. So this kind of cartoon has such levels of meaning, and I'm going to explain what all that is. Okay. So let's start for the moment. So it's true. So if you want to know the molecular biology of it, there's another entire lecture I can give about how the gene is actually expressed into mRNA. But what you need to know is, if you could, and it has been done, by the way, these experiments have been done. If you could go into a single cell and go down, let's say a bacterial cell, go down to its chromosome and zoom in on a particular locus, which is a particular gene, see what's going on, what you'll see is that there's a particular large machine which is called the RNA polymerase. Doesn't matter what it is, right? But there's this large machine that stochastically binds from the cytoplasm onto the DNA, and every once in a while, it actually starts running along like a train, reading the gene, and that's a rather fast process. And while it's doing that, as it moves along, it actually threads out the RNA, and then the RNA goes free into the cytoplasm. Now, we're not going to look at such level of detail. What we are going to assume is, well, there's at a certain sort of coarseness in time, there were no mRNA molecules, and suddenly after this thing happened, there's one mRNA molecule. That's the level of coarseness at which we're looking today. You could make much more complicated models, and the whole framework of stochastic processes, in fact, allows you to make even more complicated models. But let's just stick with this for the moment. So that means you start off with 0 molecules of X, and then at some point in time, you get 1 molecule. And then you get 2, and maybe you get 3 in quick succession, and then maybe you get 4 later, and so on. I'm drawing it like this because this is a stochastic process. The cell cannot control in a detailed fashion when this RNA polymerase is going to bump into this bit of DNA and generate something. But if you took chemistry, you would be given the same cartoon where there'll be some sort of number alpha associated with this arrow. What is alpha is a rate. For example, molecules per unit time. CULES. If you were doing chemistry, this is the kind of thing. So as a chemist, that's your intuition when you see a cartoon like this. You would immediately say, what is the rate associated with this edge? And then you would write down an equation. You would say dx dt equals alpha. Okay, some constant, for example. I mean, alpha could vary in time, but just keep it simple. There's some constant rate of synthesis, and the total number of mRNAs, assuming for the moment they don't decay. The solution to this equation, of course, is that X will increase in time. That's the deterministic solution to this differential equation. Right? But we know immediately that the whole picture is a fiction. And we know that, for example, because you can't have fractional numbers of mRNA. And you have 0, you have 1, you have 2, you have 3, and so on. So what is the connection between this type of description and that one? Okay. And there's many intuitive ways to look at it, and I'll cover a couple of them now. So one connection is so any guesses? So basically what I'm saying is, in school when you took chemistry, you would have been shown a cartoon like this, or very similar ones, where you think of DNA gene expression as a chemical reaction, and chemical reactions are typically represented as ODEs with some rates or rate constants. And these things then end up generating curves, deterministic curves. Whereas now I'm telling you that this process is stochastic. These are the kinds of curves that actually exist. So were they lying to you, or is there some sense in which these two things can be related? Yeah. Very good. So let's start talking about, let's say there's some sample time, t star. Okay? I could run the same stochastic process many times. And I don't have enough colors to do this, but basically, again, I get one of these jungle of curves. Now, one sense in which this deterministic picture of yours did capture something important and interesting about the system is that in many circumstances, not always, but in many circumstances, the average, the expectation value of x over many independent runs actually tracks the deterministic expectation, the deterministic equation. In other words, you're not looking at x of t in a single cell, but you're looking at the ensemble average of x of t. Means you run 1,000,000 cells, and then at every point in time, you plot the average of the number of mRNAs per cell. So that's one way in which the chemistry is still correct, even though there is an underlying stochastic description. It's correct to the extent that it's predicting the ensemble average. Okay, now just for those of you who are getting worried about things, this is not always true. This is only true if the thing on the right-hand side happens to be a linear function of all the x's in your system. That's one of the complexities of biology is everything is nonlinear, and so what I just said is actually only true approximately. But let's not worry about it for now.
Sorry, that distribution that you did there must be, I mean, it stops at 0, right?
Yes, it stops at 0. So what is the form? Okay, so what? Okay, good. Good. So he's pushing me to go forward. So he's going to ask me, what is the form of that distribution? So let's calculate it. It's actually fairly easy. So how would you calculate this distribution? Remember, by the way, once you can, for Markov processes, once you can calculate distributions, you can generate curves. So how would you calculate this distribution? What is the actual distribution of x at a certain time? So now the way I like to think about it is slightly, let's again turn the tables on you and think of a slightly different framework for the same thing. Let's not keep track of the curve. Let's go back to this heads, tails type of, let's keep track of when the mRNA molecule was created. Right? You can imagine, like popcorn, and imagine this thing makes a sound every time a new mRNA is created. Right? So every time you run this, you get different pops. And in general, the pops won't happen the same way if you rerun the experiment. Okay? So what we've done now is converted the question of how many mRNA molecules there are to asking how many pops there will be over a certain, let's not call this T star, let's call it capital T. I don't want too many superscripts. So over a certain window of time, how many pops will you get? Always remembering, in fact, that's exactly the number of mRNA molecules you're going to have if there's no decay, right? Always remembering that this is a random or a stochastic process. So you won't get the same number every time. So the answer to this question cannot be a number, but it must be a probability distribution. So some of you might know this distribution. It's a rather famous one. It's a Poisson distribution, right? Was this covered? Yeah, good. So what we are doing here is taking a long time window, an event that deterministically has rate alpha. In practice, because it's discrete, there'll be a discrete number, and because it's discrete, that number will be random. It'll vary from time to time, and we're asking how many there'll be. And there are many ways to calculate this, and since you've done the Poisson calculation, I won't bother going into the details of the calculation, but by definition, that's what it is. So what happens is, suppose t is some big, very large number. Right? If t is a very large number, and you would expect very close to alpha t events happening in that bin. Okay, that's the other sense in which your school chemistry was not lying to you. Sense number one is the average might represent the average of the true underlying distribution tracks the deterministic variable, even if the underlying distribution is rather wide. Sense number two is, in fact, the underlying distribution itself is very tightly spread around the deterministic quantity. And that typically happens in the large number limit. Okay? So So there's various large number theorems that prove this. So we start with that point, and then we say that with a large time window, the bigger you make the time, the more correct you'll be that actually there are just exactly alpha t events that happened in question.
So specific to this example and also many other processes in biological systems, there are feedback loops. So it is not infinitely going in the rate alpha.
So we're going to spend the rest of this class talking about more complicated things.
Okay.
Yeah?
One question. So that alpha t must be an integer, right?
Sure.
Yes. I mean,
Alpha t. So I'm not doing a rigorous calculation over here. So I just want to give you guys alpha t in one sense is the expectation value. Okay, so it's still the average, and that doesn't have to be an integer, right?
Okay. Okay.
But if you want it to be, it could be the closest integer. I mean, it doesn't really make a difference to the derivation. But when t is big, it's a very large integer. Okay? Yes?
When we are doing the stochastic simulations,
Yeah.
so other than calculating the average trajectory, the individual trajectories, do they have any other use like or significance? I mean, like
Individual trajectories are the thing. Okay? It's everything. Nobody is ever interested in these probability distributions. You're going to ask me if there's going to be an earthquake. You know, of course there are probability distributions, but the actual process is an individual trajectory. So this is what I was trying to say for the first half hour of the talk, is that often stochastic processes are taught as a way to calculate probability distributions. No, the focus is on individual squiggly trajectories because that's what's real, that's what's seen in an experiment. It's the most important thing, in my opinion. Okay. There was one last question.
As we are taking here in all these two cases, at time t tends to infinity and whatever we are getting a distribution, and the distribution is coming as here sometime Poisson, sometime Gaussian distribution and well-defined distribution.
Oh.
So every time is it expected that in stochastic case we will get a well-defined distribution? If I don't get a well-defined distribution, how do we tackle the problem?
So, fine. You've asked a detailed question. Let me address it a little bit now and move on. There are some stochastic processes where the rules of the game themselves change over time. Okay? But things like alpha and whatever the underlying rates or the diffusion constant or spatial variables can vary in ways that you can't control in their external parameters, right? So of course in those cases you have to feed in all that information. There's another situation which is problematic where you don't converge to a distribution at infinite time. You do converge to a distribution at any finite time. Okay? But that's not a problem. I can deal with both those cases. Okay? There is a very simple case, which is the case we're typically taught in college, where you converge to a so-called stationary distribution. So after a sufficiently long time, the distribution doesn't change. Now this is actually hiding what's happening inside. So although the distribution doesn't change from one time point to say the next, right? Actually individual actors are doing something very complicated underneath. So all these cases can be covered. It's not a problem. And I'm picking a particularly simple example over here, but we're going to get to more complicated things. So this is the total number of events that you get over here, right? But actually the events are happening in some discrete sense. So, sure, but yeah, you've derived all that. But what I want you to understand is the following thing. I can take a large number of these ticks here, and I know the number because it's alpha t. And I can ask a new question. I can say, in a small window of size dt, what is the probability that you get 0 or 1 ticks? And then you play the usual game where this time window is sufficiently small that you don't get 2 ticks in any window. Okay? So what is the probability of getting a tick? And by tick, I mean a particular event which corresponds to the creation of an mRNA, and so it makes a sound like a popcorn or a clock. What is the probability of getting a tick in time window t to t plus dt? This is a proper operational question because dt will be some small but finite object. Okay? It's actually very easy. You know that the total number of things here was alpha t. That's all you just divide down. So the size of the small dt interval of total is just dt over t. and you multiply that by the total number of ticks in the whole thing, this thing cancels. This is what you get, right? And of course, the result of the total number of ticks in here is Poisson distributed. I don't want to focus on that for the moment. What I want to focus on is this idea that in chemistry, you're always taught that some things are rates, which is the rate at which some continuous thing accumulates over time, number of molecules, moles per liter, various things, right? You can use different units, but it'll be some rate. In stochastic processes, the very same object, no messing around, that alpha is now reinterpreted as a probability per unit time that some discrete event happens. Okay? So this is, again, a little bit of magic here. Superficially, these two things look like very different objects. Yeah? And indeed, their interpretation is very, very different, but it's the same numerical value that's associated with both of them. So if you're creating something at a rate of 5 molecules per second, then in a very small time interval of, say, 1 microsecond, the probability of having one event is 5 times 10 to the minus 6. Okay, so we've gone from rates to probability per unit time. And today, I'm not going to bother with this whole notion of deriving this distribution. So some of you who've studied this, you can now actually break this into little bins. And in each bin, the chance of getting a heads is alpha dt, probability of a head. The chance of getting a tail is 1 minus alpha dt. And so this then breaks down to your standard sort of highly biased binomial distribution, which converts to a Poisson, which you all know, it has a formula, it has a mean, it has a variance, and so on and so forth, right? So in fact, let's write that down. So in fact, the probability distribution of the number of x's in a big interval of size t, actually e to the minus expectation value expectation value to the x over x factorial. This is a complicated thing. I'm not interested in deriving these formulas, okay? You could learn how to do this, you could look it up in a textbook and so on. I want you to understand the intuition behind this. So we've gone away from the idea of a rate to an idea of a probability per unit time for discrete events. And then you use all your, you can either derive these things or ask a friend or look it up in a textbook or use a computer and generate a stochastic process. It's not that scary. Okay, any questions? So now let's use this whole idea to start to analyze what everybody's craving for, obviously, slightly more complicated biological system. Okay, and by slightly more complicated, I mean, I'm just going to add a decay process by which the mRNA molecule gets destroyed. Right? Very simple. So then the equation, the chemistry, if you were a chemist, then this is how you would write that equation. Okay? Already you see slight complexity over here. The rate of synthesis is constant, right? But the rate of removal is proportional to the amount there is. This is like radioactive decay. Because each molecule has a constant probability per unit time of decay, just like a radioactive nucleus. That's where this comes from. And now, instead of having just a bunch of ticks corresponding to creation, which we could write as up arrows, right? You get another bunch of ticks which correspond to decay events. So down arrows. But, little googly over here, the little complexity over here is that the probability per unit time of getting a down arrow depends on where you are in the first place. Okay? And this is just the starting example of having these parameters, these rates, which in textbooks they call propensities, varying over time. They could vary over time because of some external change. They could vary over time because they depend on internal variables which are part of your system. Okay? As soon as I add this little thing, this kind of calculation just goes out the window. And that's part of the reason why I don't like these very clever derivations of Poisson distribution and so on. That doesn't generalize. Your standard trick of deriving Poisson distributions doesn't tell you at all how to deal with even a slightly more complicated stochastic process. So it's not, in my mind, a useful pedagogical tool. It's not even useful in principle, because you can always look these kinds of things up. So how do you deal with something like this? So now you have probability of uptick, and similarly probability of down tick will be gamma x dt. It's a bit of a strange thing, because that x is the actual x at that time. It's not the average x at that time. It's not the expectation value of x over a bunch of things. It's the actual x in that system at that time. And to deal with things like this is why people have created this big machinery of the master equation, which again was covered, and which I'm going to use. So I said 20 minutes, so I'll use 2 more minutes to cover this and then get back to the central aspect of the talk. So how do you deal with something like this? Well, you go back to this question. You say, now, I can't do bespoke, I can't do very specific tricks like deriving Poisson. I sit and think about it and say, well, you know, I was taught if I want to generate a curve corresponding to some stochastic process, in this case mRNA synthesis and decay, what I need to do is to find a way to calculate these conditional probability distributions. If I know where I'm starting, where am I going to be after a certain amount of time? If I can calculate analytically or otherwise probability distributions like this, then I'm golden. I can use a computer to generate the whole curve. So the way you calculate these probability distributions is now, in fact, pictorially very explicit. You think of a bunch of bins which correspond to different values of x. So let's say n minus 1, n plus 1. Can everybody see here? Is that fine? Okay, I love these long words. So these are the bins that I used to represent as the vertical axis. All I'm doing is taking that, putting it on the horizontal axis, okay? So these are the bins on top of which these histograms are sitting. Histograms generally drawn as these curves, right? That's what's going on here. And I'm interested in calculating, knowing that I started at some value of x, what distribution I get after some amount of time. And the way to think about this is, again, think about it very operationally. Imagine doing an experiment in a test tube where there are, say, 10 to the 8 cells. Right? So there's hundreds of millions of cells. And instead of computing an abstract probability distribution, I keep track of the actual histogram, the number of cells in the tube that have 0 mRNA at a given time, or 1, or 2, or 3, or 4, or whatever it is. Right? So I associate a number with each of these. Question? No. So messenger RNA is not converted to protein. The messenger RNA floats around. And while it's alive, the ribosome will translate to protein. But a single messenger RNA can actually be used to template 10, 20, even 100 protein copies. There's actually an enzyme that chews up the RNA. There's a separate enzyme that binds to one side of the RNA and destroys it. It's literally broken apart. Okay, thank you for that question. I didn't mean to. So we haven't never talked about protein so far. So his question was, does the decay correspond to an RNA being changed to a protein? That's not how it works. Okay. We can discuss that later. Okay, so I'm going to associate a number here, which is P0, P1, P2, Pn minus 1, Pn, Pn plus 1. And these Ps literally represent the number of cells, 100, 2,000, 755, whatever, the number of cells in your tube that have this many RNA. Right? And in principle, this could go on infinitely long. Now, I think about a certain time point. And I say, at time point t, I had some p n. For all n, Any n you give me, I tell you how many cells there were at time point t, right? And I'm interested in how many cells have that number at some later time point. Slightly later. Okay? But this is an actual number, okay? This is an actual finite time. It's like a millisecond. Okay, this is like 50,000 cells. Maybe there's 52,000 cells. Okay? So how do I make this leap in time? And that's the whole framework of the master equation which you saw yesterday, and I'm going to explicitly put it down here because I'm going to use this in the second half of my talk. So, yes. They're not the same. They're not the same. I need to understand how to get that from this. Same n. How many cells have 65 mRNAs at time t? How many cells have 65 mRNAs at time t+1? Just as an example, okay? So, how do you do this? Well, you do it just by staring at it. It's actually fairly straightforward. There's a number of cells here, which is Pn-1. There's a number of cells here, which is Pn. There's a number of cells here, which is Pn+1, and so on. The heights of these bars show you the number of cells, the actual number in here. And we know something already. We know the probability that a cell gains an mRNA in a single cell, in a short span of time is alpha dt. And we know the probability that a cell loses an mRNA in a short span of time is alpha x dt. In this case, the x is actually now being written as n to make it explicit that it's sort of an integer. Okay? So, how many cells will actually make the leap from here to here in that short span of time? We don't have to worry about stochastic processes at all now because we know that there are hundreds of millions of cells here, so I can just report the averages. Okay? The number of cells that are going to go here is the total number of cells you have. Then each cell has a certain probability of leaping to the right. You could also imagine a cell losing an mRNA. And again, it's proportional to the number of cells you have, and then the rate per cell, which we've also calculated. So in this case, it'll be called gamma n dt because I'm representing the number as an explicit integer, not this x anymore. Okay? Forward one will have Pn. If there's 50,000 cells, the number of cells that move to the right will be proportional to 50,000. If there's 100,000 cells, this is the total number of cells that made the transition in a short span of time. In a single cell, it doesn't. I'm asking, there are 50,000 cells here. Each cell has the probability alpha dt of going to the right. So how many cells in total will move to the right from here to here? That's the question I'm asking. It's very important to realize that that p has to be there. That's the point of this exercise. I'm not modeling a single cell. I'm modeling the entire histogram. You're interested in a single cell, and I direct you to this part of the board where once I calculate the histogram, I can do magic things about single cells. Okay? This is always the confusion in stochastic processes, where people are always spoon-fed the histograms and then left to themselves to figure out what single cells do. That recipe is to figure out what single cells do. I'm only calculating the histogram. Okay? But there's a couple of other things that can happen. Not only can cells in this bin, leave, there's other cells that can come in. And we can calculate that in exactly the same way. Right? And the only complicated thing about this is that there's an n+1 here. Okay? So this is alpha dt, this is alpha dt, this is gamma n dt, but this is gamma n+1 dt, because every cell here, gamma. Okay? So now we can finish the calculation. Right? So we know that the number of cells in the next time point is the number of cells there were minus the number that went away plus the number that came in. And so let's write that down. It's minus p n alpha dt plus gamma plus Pn gamma n dt. That's these two arrows. And then it's plus Pn minus 1 alpha dt, is this arrow. And it's plus Pn plus 1 gamma n plus 1 dt. That's this arrow. And when people first see this, they get a bit confused because this is a decay process, but it comes in with a plus. What is going on? Nothing complicated is going on. I'm talking about the number of cells here, and that number of cells will increase because cells here lost an mRNA. That's all. Yes? We are looking at the total number of cells in this bin. That can change because a cell here lost an mRNA. It can change because a cell here gained an mRNA. But what we are calculating is the total number of cells in this bin. A cell will jump from here to here, yes. And therefore, this bin will go down. Okay? Good. So the reason I say this repeatedly in so many different ways is because it is a complicated thing. Okay? It is something that requires a lot of getting used to. This way of thinking. This way of jumping back and forth between what a single cell is doing and what the whole histogram is doing. Okay? It's not easy, it's not obvious. And I do urge you to go back and read notes. There's a lot of material about this stuff online. I'm giving you tools so that you can access such notes. Okay? Good. So this is the equation. And let's just finish it off. So obviously then I can do the usual calculus trick. I separate the left and the right in the following way. I subtract the current time point. I subtract the current time point from the next time point. Right? I divide by dt because everything on the right is proportional to dt. And then I write down what's left. Okay, so this is it. Yeah. Right. This is where we made that assumption. We made the assumption that dt was so small that you can't have two events happening in a little bin. This is where that hidden assumption came in. Okay? Good question. Yes, Vijay. Okay. Now, I'm not going to help you solve this. That's not the point of this exercise. What is this, by the way? So the left side is dp and dt. By the laws of calculus, the left side is dp and dt. Everybody fine with that? Okay. So what do I have here? I have an infinite set of differential equations, a separate differential equation for this bin, this one, this one, this one, this one. And if that wasn't bad enough, these equations are all coupled. What are the variables here? The variables here are no longer the number of mRNA. That's not the variable in this picture. The variable in this picture, the thing that changes over time, is the number of cells in each bin. And I have a separate differential equation for the number of cells in bin 2 or bin 5 or bin 67. But even worse, the differential equation of bin 67 depends on bin 66, on bin 68. Okay? So we suddenly realize how complicated stochastic processes are. You went from a single ordinary differential equation, okay? That's it. To this crazy beast. One variable, infinitely many, nothing coupled about this. Every variable is coupled to things above and below. Okay? There's only one thing that is sort of a silver lining here. I don't know if you can see, but at least this differential equation is linear in its variables. Okay? In principle, I could make this a function of x. In fact, I could make this also a function of x. I could make these things complicated nonlinear functions of x. Right? And that makes the ODE sort of difficult to solve. What would that do? That would just enter as alpha and gamma becoming functions of n in this formulation. But those things are just coefficients. And in fact, those are constant coefficients because for any bin, the n is constant. This equation is still linear in its variables of interest, which are the pn's. Are there any questions about this? Yes.
So I lost the intuition when we go for the derivative.
Just repeat the last point. You lost the intuition for where?
You went for the derivative of the...
Oh, that's just a trick. This part of it is fine with you? Yeah? All I did was subtract this from this and divide it by dt. It's just a trick.
We were trying to answer...
Yes, yes. I get your question. I'm going to answer it. Now that I have this equation, okay, if you're very clever and work very hard, you can put down Pn of 0 is like P0 is 1 and all the other. Oh, sorry. Let me just step back 1 second. One last thing. What were these Ps? These were the number of cells in each bin. And the total number of cells is like 100 million, right? But now I'm going to do another thing where I divide everything by 100 million. Because this is linear, this gets divided, this gets divided, this gets divided, then nothing changes in the equation. And suddenly I went from an operational and actual histogram, now I've gone to a normalized probability distribution, okay? So far. So just bear with me for a second. So what I now have is an equation that tells you how the shape of the probability distribution changes over time. Right? It's amazing how much I managed to slide into this little discussion. I made assumptions that all of you agreed with at every step. And suddenly I come with this extremely powerful thing, and that's when people are taken aback and said, really, can we derive this? We did. We just derived an equation that tells you how the shape of a full probability distribution changes over time. There's only one last thing to do, which I'm not going to do, because this is the hard part. I take the initial condition as P0 equals 1 and everything else equals 0. Meaning initially, all cells have 0 mRNA. I plug it into this thing, and then you can either do it by hand or in a computer or what have you. You can then calculate Pn of t, which is exactly this object. Okay? So I've just taught you a way for a rather complicated stochastic process. It looked simple to begin with, but it's complicated. For a rather complicated stochastic process, I've given you a recipe. I haven't solved it. Right? It takes some skill to solve equations like this. To solve it means, given an initial condition of the p's, to give me the p's for all time. Once you solve it, you get this. Once you get this, you can use a computer to get this, and then you're done. Question?
As we have seen the master equation, and there the graph of x and t also in the left board.
Yes.
Yeah. So at any finite time, what the distribution of x was. So may I tell that the distribution evolves following the master equation?
Yes. The distribution actually tells you how this thing changes over time.
And the second question in this regard is, as the form of the master equation is general form,
Yes.
or whatever with stochastic process we start with. Yes. So can I tell the final distribution only depends on the transition rate?
So I don't want to get into the issue of whether this converges to a fixed final distribution. Right? So for example, if I remove the gamma part of it, this doesn't converge onto a fixed distribution. Okay? There are many ways to sort of solve this, right? But the real way to solve it for stochastic processes is to calculate conditional probability distributions, not just from starting at P0, but from starting at P4 or starting at P67, because such decisions are things you'll have to make when you generate this curve. Okay? So it's a mistake to think that the whole point of this is to calculate the steady state distribution. It's not. The whole point of this is to calculate conditional distributions from any initial condition. In some simple cases, there are steady states, and those steady states are, you can write them down in simple forms and all that, right? But those are aberrations. They don't help you in real life. Don't worry about it. That's sort of math Olympiad type of stuff. It doesn't ever come up in your research. Right? Okay. Good. So in the remaining 15 minutes, I'm going to set up the question that we're going to solve in the next half of the talk using this machinery. I'm actually going to erase a lot of this stuff. So are there any questions about it? Maybe I'll leave this part of it and start erasing on that side. Are there any questions? Look, again, different people have different ways to solve this, right? You can use Laplace transforms. You can just make this a big matrix, right? Because it's linear, it's an infinitely large matrix, and then exponentiate, right? So in the next half of the talk, I'm actually going to give you a specific example of this where we're going to solve this, and we'll see what that looks like. Okay. There are many, many ways to solve it. One very popular way is to actually discretize is to imagine that the difference between two x's is rather small compared to the total x, right? Then you'll notice that not only do we get a time derivative here, which is like p of t plus dt minus p of t, but you actually get a sort of spatial derivative, which is like p of n plus 1 minus pn. And that's the move that you make to get a Fokker-Planck equation, which you can write down as a Langevin equation. I'm not getting into that today. I'm not getting into methods to solve. I'm getting into the intuition to set up the problem. Both are important, but I think this is much more important the first time you see it. Okay. So if we're okay with this, I'm going to erase all this stuff. I'm just going to leave maybe that part of the board. And tell you a little mystery. This is actually a research project we started and finished a couple of years ago. Where's Sanjay? Inspired by conversations I had in Delhi on bacterial growth. And if it hadn't been for exposure to things like this, I wouldn't have noticed there was a research question inside. So part of what having exposure to stochastic processes allows you to do is not simply solve very, very complicated equations. It allows you to go inside and realize there are interesting dynamics going on. It's a point of view. I don't care if you cannot solve these equations. It's not important because you have computers. Okay. Is somebody going to talk about Gillespie, or you already, not necessary. There are other ways to solve these things other than this very complicated way. And we can discuss that maybe over tea or coffee. Okay. So I'm going to tell you some classical experimental results in microbiology. How many of you have ever grown a bacterial culture? Wow. Mouth pipetted? No. So, accidentally grew, in your fridge. Yeah. So when you do bacterial growth, it's rather amazing. So when you walk into a lab where there's a microbiology lab, what you see is a beaker, and initially you put in, well, let me actually show you the most magical thing that you'll see in the lab. There's a big black box. This is sugar and salt, plus water, right? There's a tube, and it's pumping in sugar and salt and water into this. Right? And there's a pump that's causing the pumping. There's another tube pulling stuff out of this, right? And what you get over here are living cells. Right? This is the most ridiculous thing, right? So I'm pumping in sugar and salt and water, and I'm getting out a steady stream of bacterial cells. Right? This should surprise everybody. What is going on in here? Right? Obviously, what happened in here was somebody put in one bacterial cell in the beginning. And if you did that, you're going to get more of them. And if you didn't do that, this will never happen. And if I change the species, I get a different species. It's not that I can put E. coli in here and get bacillus outside. Right? So this is the magic of biology. It takes substrate, which is non-living, purely chemical stuff, and it makes this amazing state of matter, which is the living state. Now let's not look at this experiment. This is called a chemostat. Whoops. Let's look at a much simpler experiment, which is that you take your flask, you put in sugar, salt, and water, you inoculate with a certain number of cells, and then you watch how many cells there are over time. The cells grow and divide. And there's a couple of ways in which you can do this experiment to track. How would you measure how many cells there are in here? Okay. A quick and dirty way to do this is to simply shine light through here, and you measure the absorbance. And typically, you measure it 600 nanometers. So this is called optical density at 600. Happens to be a wavelength where bacteria will scatter light, and you get nice measurements of how opaque this culture is. So OD600 measures the total number of scattering objects of a certain size in this beaker. Okay? And if I plot, let me do it this way, time, I plot OD600. In fact, I plot the log of this, and you'll see why in just a second. Everybody knows what we're going to get, right? What is it? Straight line, more or less. I mean, leaving aside the sort of initial lag and the final thing, but mostly you get this, right? That's the most important part of what's going on here. And what is this called? So, so very confusingly in biology, it's called both exponential growth and logarithmic growth, right? So it's actually exponential growth. It's not logarithmic growth. Right? The other confusing thing is that cells divide and they multiply, but what they're actually doing is increasing in number. Okay? So, why do you get exponential growth? Have you ever thought about this? So why? There is an obvious answer, there's a non-obvious answer. Let's start with the obvious one. Why do you get exponential growth in the number of cells in a tube? Well, you know, one cell makes two and that's it, right? So it's, two cells make four. So in fact, you get geometric growth, which is sort of, at this scale of things, looks like exponential growth. Okay, that's fine. But what was very mysterious to the first people who worked on this, and Campbell and others, the idea was that they looked at these curves and you could actually get this thing going like this. For 24 hours, you would get exponential growth at a fixed slope. Okay, the slope to the sensitivity of the measurement did not change here to there. And did not change in Bangalore or Chicago or anywhere else. Right? And so this is one of those things where biology suddenly became quantitative. There is a slope. And that slope is associated with the composition of the medium and the species of the bacterium, temperature and so on, but nothing else. Importantly, it doesn't change over time. And what people realized this meant was a very important thing. It means that, so imagine you track a bacterial cell, it grows, a little septum forms, and then you get two bacterial cells. Right? And these two, of course, are simply fed back into the system. Each one becomes a post-division daughter cell, and it grows and divides. Okay? The fact that this line is constant slope over many, many hours, and anywhere in the world, actually means that the chemical composition of the immediate post-division cell is not changing over time. Okay? Of course, the composition of a cell, as it grows from an immediate post-division cell to just pre-division and then divides, of course, a lot of things are happening in the cell. But if I synchronized and I just tracked what the composition was of immediate post-division daughter cells, that composition doesn't change over time. That's the inference of this. Because if it did change over time, all kinds of things would go crazy with this curve. Okay? So there is a very simple explanation for exponential growth. Now what you can do is add an antibiotic. Okay? There are many, many antibiotics. You know what they are. Penicillin, erythromycin, kanamycin, whatever it is. Right? There's a lot of things you can choose. What do you think happens to this curve? You redo the experiment with antibiotic. Starting with the same value. This is log, right? So this value, 0 doesn't matter for log. Right? It can be positive, negative. So what happens with antibiotic? I put a little bit of antibiotic. Anything will grow with a little bit of antibiotic. Okay, how many people think the line will still be straight, but the slope will reduce? Okay, how many think something else will happen? Like what? Right, you've been taught that antibiotics kill bacteria, right? So really this should not go up at all, but if it's just a little bit, something should happen, right? So actually, the first person who said it was right. You get a different curve, but it's still a straight line, right? In a log-lin plot of OD versus time. Right? But if you start to look here, you'll start to see some deviation. Okay? There is a problem with OD. The problem with OD is it cannot distinguish living cells from dead cells. Anything that's around and scatters is going to pick up. Well, we'll get to it. So you're trying to make a connection which I'm going to make in the second half. We're going to take the coffee break and come back and finish the story. But just hold on to that thought, okay? So what I'm going to tell you is OD 600 is a great way for an engineer to measure the number of cells in the tube, and most of the time it's okay, right? But you'll start to find that as you do perturbations and complicated experiments and so, you want to be able to distinguish living from dead cells. And this technique does not let you do that. But there is a technique that was invented a very long time ago that very nicely allows you to do that. What you do is you take a little sample out of this thing and you put it into a petri dish. And this petri dish doesn't have antibiotic. It's goody, lots of sugar and salt and very nice growth medium, right? Rich medium. You take a little sample of this and you dilute it and you pour it onto this dish. And you put that dish at their favorite temperature, for example, 37 degrees for E. coli. You come back the next day and you count the number of colonies that were here. Right? What are those colonies? Each of those colonies started out as a single bacterium because you took such a dilute sample of this thing. Okay? It's a single bacterium that grew into a colony. And this way you don't need a microscope, you don't need anything like that. So just with a fridge and an incubator and so on, you can count the number of living cells per ml in this thing. Okay? And that thing is called CFU per ml. It's called colony forming units. So before I get to the OD, in fact, I should have started with CFU, and I'm going to put log of CFU per ml. And remember, CFU per ml is living cells, which are called C sub L. Whereas OD 600 is C sub L plus C sub D, living plus dead cells. The cells don't lyse, they'll continue to scatter light. You will pick them up. So now let me show you something interesting. This is a fact. As you add more antibiotic, for CFU, you still get a perfectly straight line up to the precision of the experiment. What if I add even more antibiotic? No. I'm going to put those on the same. I just changed the axis here, and I'm cheating by using the same curves. Right? I'm going to put them both on the same graph in just a second. So what happens? I add even more antibiotic. What happens? Well, okay, fine. So maybe you get something like this. Add even more antibiotic. What happens? How many people think it just stays flat? How many people think something else happens? What happens? We're not running this long enough that we're going to pick up a small number of mutants which are going to spread over the population. You'll be discussing issues like that later in this school, but we're not discussing that now. This is just a few hours. Okay? We're not picking out the resistant clones inside this population. This is a general bulk population. What happens if you add more antibiotic? What is this? It's the number of living cells per ml. And you started off with 100 million, or let's say 10 million, or whatever it is. But you started off with some large number. Now add more antibiotic. What do you expect will happen? Goes down. Now how will it go down? It will go down, right? Because the number of, somebody said, the cells die. You were taught in school, antibiotics kill cells. So let's take this word kill seriously. The cell died. So what? It goes down, okay? What will be the form of the decreasing curve? Linear. It's your guess. You're right. Experiments were done in the 70s. More antibiotic, this is supposed to be a straight line. I'm sorry, it's a bit curved. More antibiotic. Yeah, up to the level of sensitivity of the measurement, right? Maybe below some number of CFU per ml is, you can't really measure it very well, right? These are straight lines that cover several orders of magnitude, powers of 10. Okay, so this is the setup. In the last 2 minutes, I'm going to ask, yes? Yeah, this is a fixed quantity. This is value, whatever. So it's true that if you wait long enough, the bacteria will chew up all the food, right? And then this curve will change. I'm restricting myself to this very large and robust linear or exponential growth regime. The kinds of things you're talking about will happen. And incidentally, if I suddenly shift the antibiotic level, there'll be some sort of transient. So I'm talking about the large asymptotic behavior in the middle, after the transients, but before you run out of food. If you want to do that. But this is actually stuff you can do in a tube. So let me now, before we break for coffee, ask you. Now, exponential growth, you all agreed, it was explained in sort of a school level explanation where one bacteria makes 2, makes 4, makes 8, and that's why you get exponential growth. How do you explain exponential decay of bacteria? Pure exponential decay over several orders of magnitude over several hours of an experiment. As if it was a radioactive nucleus. What are the implications of this? I want you to just think this out. And at precisely 11, I don't know if there's a clock here, but it will stop. As soon as you unpack some of this stuff, and in the second half, we're going to figure out what's going on. Can somebody see there's something deeply mysterious about this? You can explain exponential growth by geometric growth. Exponential decay means some cells died this time, some cells died here, some cells died here. They don't all die. I mean, you could have imagined that it was something like this. I mean, you could have imagined. And there are situations where, for example, cancer-related deaths in humans, if you plot something like this, right? Nothing happens for a while and then everybody dies. You could have imagined it was something like that. Perfectly valid guess. But that's not what happens. Right? So this is the mystery. It's been known since the 1970s. That you can set up a system, and if you add enough antibiotic, they display exponential, pure, to the precision of the experiment, exponential decay over several orders of magnitude. The last thing I want you to think about is, what is happening here? Okay, so what it looks like is there is a stochastic process with a rate corresponding to growth and a rate corresponding to death, and each of these things behave like fundamental particles. Right? But that's not true. Bacteria are not like that. Okay, so now we're going to have to decide whether we're satisfied with this type of explanation or whether there's something more microscopic going on. And that's what we'll cover at after the break, because we'll stop now. Yeah.
Actually, I wanted.
That's exactly what I'm going to get at. Thank you.
So those counting colonies, how is that? I mean, you're counting on the millions, or what are you doing?
So what you do is, you, in practice, when you do this experiment, you take a sample from your tube, you put it on a petri dish, but then you take a factor of 10 dilution and a factor of 100, and you do a series. And you'll find that the first few of those are completely covered, last few have no colonies, and somewhere in the middle you get a finite number which you can count. And then you invert the dilution factor to measure the number.